新冠治疗新药。
One is biotech and microbio Shanghai International Press Conference on Phase 2 study result for SNS812, a pan-COVID new drug, now begins. International investors and friends of the press here in person and online, good afternoon, everyone. Thank you for attending the International Press Conference on Phase 2 study result for SNS812, a pan-COVID new drug co-hosted by Oneness Biotech and Microbios Shanghai. Oneness Biotech was notified by CRO last Friday that the Phase 2 clinical studies data for SNS812 has been unblinded. So we will be explaining in detail to our investors and the international market the efficacy, safety, and international competitiveness of SNS812. First, let me introduce our guests and the organizer. First, sitting in the middle is academician of Academia Sinica and Oneness Biotech Chief Scientific Advisor, Professor Yang Panchi, Principal Investigator of the study and Professor of NTUH Department of Emergency Medicine, Professor Chen Shiying, Virologist, Professor of Department of Clinical Laboratory Sciences and Medical Biotechnology at NCTU CM, Professor Zhang Shuyuan, and organizers Dr. Zheng Shuling, President of Oneness Biotech, and Dr. Zhang Yizhong, President of Microbio Shanghai. Co-developed by Oneness Biotech and Microbio Shanghai, SNS812 is the only siRNA drug that can treat COVID-19 worldwide and has completed Phase 2 clinical study, successfully proving its safety and efficacy in COVID-19 treatment. Relevant data will be shared in detail with everyone in today's press conference. First of all, we would like to invite President Zheng Shuling on stage for a short remark. Our friends of the media, in person and online, distinguished guests, friends from the press. First, on behalf of Oneness Biotech, I would like to express my gratitude to everyone participating in today's international press conference. I am honored to announce that the Phase 2 clinical study for SNS812, a siRNA new drug for COVID-19, has achieved significant efficacy and well-supported safety outcomes. We hope that it will become the best option for worldwide COVID treatment based on its safety, precision, and efficacy. This clinical study is randomized, double-blind, and placebo-controlled. It was conducted according to FDA's guidance and international standards. And all 135 participants were asked to receive inhalation treatment at the hospital with complete viral sequencing. Analysis indicates that all viral strains fit into the 8 most globally prevalent genotypes, and the JN.1 variant was found in 50% of subjects. The final results of the study show that SNS812 is not only safe for human use, but is also the best clinical outcomes compared to currently available treatments regarding both the duration from testing positive to negative and resolution of all target symptoms. Notably, it showed statistically significant clinical results versus placebos in treating the following symptoms: those affecting the central nervous system, such as loss of taste, loss of smell, and those that are precursors to severe COVID, like shortness of breath, fever, chills, and more. There is currently no known medication that treats different COVID strains both safely and effectively. Many currently available drugs and vaccines have multiple side effects and are rendered useless due to the inability to adapt to the speed of virus strain mutation. The results of this study for SNS812 are not only merely a milestone and success of Oneness Biotech and Microbio Shanghai, but also a medical breakthrough on an international scale. We are confident that SNS812 will be a valuable new drug that prevents the world from being burdened by COVID-19 and promotes the return to daily activities. In the future, Oneness Biotech and Microbio Shanghai will work together with international partners through licensing collaboration for SNS812 so that this groundbreaking medication may receive EUAs and approvals for targeted countries or regions. For today's press conference, we invite Dr. Yang Panchi, an academician of Academia Sinica, who is Oneness Biotech's chief scientific advisor and a physician from the National Taiwan University Hospital, Department of Thoracic Medicine, Professor Chen Shiying, the study's principal investigator from NTUH's Department of Emergency Medicine, and Professor Zhang Shuyuan, a renowned virologist, to provide overview and future trends of COVID-19 and dive into the results of this SNS812 study for better understanding of the new drug. Thank you. Thank you, President Zheng. We now invite Dr. Yang Panchi, academician of Academia Sinica and Oneness Biotech Chief Scientific Advisor, to explain the current status of COVID-19 globally and the limitation of current vaccines and therapeutics. Good afternoon, our friends. This is my disclaimer. Next, I would like to share with you the global endemic overview of COVID-19 and current vaccines and therapeutics. COVID-19 may seem like it's slowing down around the world, and people believe that it could be just like any other flu, but it's actually more fatal than flu. The mortality rate is 4 to 5 times higher than flu. According to WHO, the accumulated infection has reached 700 million, leading to over 7 million deaths globally. Even though people feel that it's like a flu now and the symptoms are mild, so for a better economy, we should not control the quarantine so strictly anymore to pursue better economy. But in fact, around the world, there are still 3 to 4 peaks of infections around the world. So it's growing serious in countries around the world. The actual infections are actually 2 to 20 times higher than what has been reported from each country. And we can see the head of the Emerging Diseases and Zoonosis Unit, Dr. Maria Van Kerkhove, commented in her interview that it will continue to transmit indefinitely. But currently we use vaccines, but the virus strains mutates very quickly, 4 to 5 times per month. So by the time we are injected with the vaccine, it has been rendered useless. So a lot of the countries and pharmaceutical companies encourage that you take more vaccines, that you receive more vaccines to increase the potency to prevent infections. But if the vaccines are useless, it doesn't matter how much vaccines you get. Even if you get 4 to 5 shots of vaccines, it's still invalid. And also, when you boost your antibody higher in your body, there are immunogenicity problems. And also, in terms of treatment, there are no therapeutics to really treat COVID-19 because we keep having new mutant variants. So in the future, it is predicted that every year there will be 110 million new infections. And according to our Vice Director of Health Bureau, Luo Yijun, has also said that even though the peak of COVID-19 and flu have passed, but the number of deaths was still record high, and a new wave is underway in September. And we can see 84 countries around the world are still worried that the COVID infection could peak during the summer. The United States was also worried that they will experience the largest COVID wave in the summer. And it's also the same for China. The currently available therapeutics, including Paxlovid, a lot of people have taken Paxlovid. When you take Paxlovid, they will ask you what other drugs are you taking at the moment, because there are a lot of drug-drug interactions, and there are many adverse events. About 70% of the incidents are related to the drugs, and other therapeutics are facing the same problems. Some could lead to more dangerous virus strains, and some could lead to mutagen concerns. Some will impact your liver functions or teratogenic concerns. So there are no optimal drugs available at the moment, especially when we look at Paxlovid. According to the New England Journal of Medicine, regarding people who have been vaccinated, they want to see if Paxlovid can lower the symptoms or mitigate the symptoms of the people who have been vaccinated and infected. And according to the over 1,000 patients looked at, we can see that there is no statistical significance in the time to the sustained alleviation of all symptoms receiving Paxlovid and placebo group. So according to FDA, even though in April, FDA approved the monoclonal antibody for COVID-19, and it's used for the subjects without sufficient immune response. But 4 months later, FDA further limited the use of this antibody because the variants have again mutated, so it's rendered useless for the new mutated variants. So even though we have new drugs coming out and vaccines coming out, but the viruses are mutating too fast, so the treatments are rendered useless. So how to really provide an effective inhibition is the most important thing. So when we look at the mutations, they are happening at a very fast speed. Every 3 to 4 months, we have a new wave of variants leading to new infections. We can see in 2024, as of May 24th, 2024, we can see this JN.1 was the mainstream, but now we still have new strains coming out. So the development of vaccines are falling behind virus evolution, even though the United States suggests that we use JN.1 variant as the mainstream for 2024-2025 vaccine, but very quickly it was replaced. So using JN.1 variant to prevent as vaccine will not prevent COVID-19 infection, because as a vaccine, it will not be able to prevent the patients from being infected with the new variants. So how to come up with the new generation of therapy is something that is urgently sought around the world. The Department of Health and Human Services in the United States initiated Project NextGen by investing $5 billion to develop therapies and vaccines ahead of virus evolutions. So they're encouraging large pharmaceutical companies to develop these therapies and vaccines that will not be limited by the viral mutations and it has to be broad spectrum to fulfill the needs. So we invite Professor Dr. Zhang Yizhong for further report. Friends of the press, good afternoon, everyone. I will now share with you the introductions of SNS812, the siRNA. It is co-developed by 1SM Microbio Shanghai. It is the only pan-COVID siRNA drug. It is a new generation of antiviral drug with precision, safety, and broad-spectrum effectiveness.
It's also a breakthrough drug to replace existing treatments. Its NOA was already published on the top 10 international journals. During the development process, it's very prudent. We go through a really scientific validation process. So in 2021, we contact the FDA for the first time to understand the legal requirement from FDA, and same year we apply for patent. We file our results. We publish our results on the international journal. In year 2022, it's our second time contacting the FDA. So we try to understand if we have a comprehensive data, and we apply for IND and got approval. So we got the green light for phase 1 study. And then the unblinding results for the phase 1 was really good. So we applied for phase 2 study application in 2023, and now we have the unblinding results. Why SNS812 is special? You can see from this chart, you can see the black bar representing the mutation rate of each genome, and the color bar means the different genome. So the higher the black bar is, you can see how a higher the mutation rate is. And all other treatments are targeting the sequence with higher mutation rate, but ours is targeting a critical location with really low mutation rate. This segment actually remain unchanged since year 2003, and that's the reason why we can reach a broad spectrum effect. It's effective in terms of a lot of highly mutated strains. So current therapies include nucleoside analog and protease inhibitor. The nucleoside analog is like in the left button of the chart. Nucleoside analog has side effects due to lack of specificity, and that's why it has higher risk of usage. And we can see the bottom right. You can see it's a mechanism for protease inhibitors. We know it's a famous Paxlovid, but it has a lot of side effects and it will become ineffective due to mutation. SNS812 can specifically target the highly conserved genome, so that's the reason for its high efficacy. So from 2022 Q3 to 2023 Q2, we conducted phase 1 study in US. The study actually can be divided into two stages. Stage 1, we do the single dose escalation from 0.3 milligram to 1.2 milligram. And the second part, the escalation actually from 0.6 to 1.2. And then we have the control group. The phase 1 study result shows that the subjects actually, in terms of their vital signs, physical examination, cardiopulmonary function, blood biochemistry, coagulation function, or urine, they have no significant abnormal reactions. All AEs are not related to the study drug. All data is normal. SN812 caused no immunogenicity and no ADA was found. So we have the result that the SNS812 is really safe without generating any antibody affecting the efficacy of the drug. Now we would like to invite Dr. Chen to explain the results of the study. Thank you. Friend from the media, and all the friends online who care about the drug development. Good afternoon. I'm Chen Shiying from NTU Hospital. Now we'd like to share the trial design and data analysis of the phase 2 study, and we'll also report the primary endpoints and secondary endpoints. The trial conducted in 2023 Q4 to 2022 Q4 to 2023 Q4 to 2024 Q3, and focus on the COVID patients in terms of its efficacy, safety, tolerability, and pharmacokinetics. And the sites are NTUH, TMUH, Shuanghe, and Wanfang Hospital. We have 135 subjects, and we will review if the patients can be treated for the first day treatment, and you need to have the COVID symptoms on the day we do screening. We need to check if they do have the COVID-19 symptoms. And then we have three kind of dosage: 100 milligram, 200 milligram, and the placebo, and we do the follow-up of 2 months. Our primary endpoints are safety and tolerability. Secondary endpoints are the time to sustain elevation and resolution of COVID-19 symptoms between SNS812 groups and placebo groups. So we are focusing on the COVID-19 patients. The BNI need to between 18 and 32. And the patients, we need to make sure the patients really have the COVID-19 syndrome within 3 days prior to administration. And we also have some requirements for the clinical trials. For example, subjects need to take measures for contraception. And we also need to exclude severe cases or the potential cases which might progress to severe cases, or patients need to be hospitalized. We'll exclude these cases, and they cannot be vaccinated, and they cannot get any monoclonal antibody for SARS-CoV purpose, either for treatment or for prevention, and they cannot attend any other clinical trials in the same month. And they need, so if the time is within 5 half life of other test drugs or 3 months, they will be excluded. The pregnant females will be excluded. And they cannot have other respiratory diseases like acute rhinitis and sinusitis. And they need to have normal cardiovascular function and kidney and liver function related diseases, which will affect the conducting of the trials will be reviewed by the investigators. If the investigator think these factors would interfere the conducting of the trials, they will be excluded. And the subjects cannot have any acute systematic infection, and HCV, HBV will be excluded. But if the subject has a history of has the active HBV, it will be also be excluded. The whole process we take really seriously. So at first, the investigator and co-investigator will explain the whole process to the patients and get the informed consent. Then we'll do the screening to make sure if the patient should be excluded. And every patient will be tested with PCR to make sure they are infected with COVID. So we need to confirm its virus strain. And we will also use a color test to make sure if a viral load is increasing or reaching plateau. because the patients might be confused or have some mistake in their memory. So we need to make sure through our test. Then we do the double blinding and randomization process. And we will ask the patient to do inhalation treatment in the hospital. Our nurses will monitor their usage of this nebulizer. And the subjects will fill in their symptom progress online. So they can do that everywhere without affecting their family time. And we value the safety a lot. So, and our definition is alleviation and resolution for consecutive 3 days. That's our definition for alleviation and resolution. So it's quite prudent, takes a higher level. And this is our randomization of subjects, excluding some other subjects with screen failure. We have uh, we have several subjects, they have their own personal issues, so they didn't join the study. Now we have 135 subjects join the trial. And you can see we have the baseline data, including the age, gender, smoking history. There's no significant difference. And the risk factor of COVID-19, also no difference. All subjects were vaccinated at least once of this COVID vaccine. Because in Taiwan, we have a higher vaccinated rate here. And we have several withdrawn subjects due to their personal schedule. In terms of our analysis, we use full analysis set. And safety set refers to all randomized participants who receive at least once administration of study drug. For modified intention to treat analysis, these population are all randomized participants receiving at least once administration, and they have received at least one time of post-baseline screen, and then they will be considered into this group. Now we will see the primary endpoint. During the treatment time, the number of AEs, we have 57, 51, and 50 with no statistic significance. You can see the SAE, the number is 0, and we don't have case which will lead to the termination of the trial. We can see the 27 patients with AEs, they have some syndromes like the headache or other syndromes, or their white blood cell variation. There's no significant difference for that. And for respiratory disorders and vomiting, runny nose, sore throat, the 3 group data, also there is no difference for that. So most of their symptoms are due to their original infection of COVID-19.
So we can see the infections and investigations, there are no significant difference between the 3 groups, including the skin and subcutaneous tissue disorders, including eyes, the heart, or any harm during the follow-up or new new tumors discovered, these have no differences. Now we can see the three groups are very consistent with no statistical difference, and there's a very low incurrence rate. And in terms of secondary endpoint, of all 14 COVID-related symptoms, when we look at all of these targeted signs and symptoms, we can look at their time to sustained alleviation. By sustained alleviation, we mean that the symptoms must be mitigated and continue to be mitigated for 3 days. That's what constitutes as sustained alleviation. So we can see the 200 milligram has a significant statistic difference compared to placebo. And sustained resolution means you have to be at least 3 days completely free of the symptoms. For example, no nasal block for 3 days. And if we look at sustained resolution, we see statistical difference in both the 100 milligram and 200 milligram groups compared to the placebo. So we have a faster resolution of the symptoms. And for time to sustained alleviation of shortness of breath, in terms of you can see difference between the 100 milligram and 200 milligram groups. And sustained resolution for shortness of breath, we can see 200 milligram has a faster speed in sustained resolution of shortness of breath. The significance of shortness of breath clinically is that it is, according to past research, it is one of the key factors for discernment between mild and moderate COVID-19. An analysis from the data from 7 studies with a total of 1,813 COVID-19 subjects, it revealed that shortness of breath is the only symptom related to severe cases and hospitalization outside of age. So we can see the p-value is smaller than 0.001, and the severe COVID-19 incidence rate is about 0.4%. That's according to 2022. It's even lower now. So there are 135 participants in this trial. You may think that it's not a significant number, but when we look at the shortness of breath improvement, it is a significant effect. So I think this drug has potential for reducing severe cases for COVID-19 in the future. And in terms of sustained alleviation of loss of smell, even though in both the 100 milligram and 200 milligram, we don't see any statistical differences. But when we look at the sustained resolution in both groups compared to placebo group, there are statistical differences, which means the time to sustained resolution is quicker. And in terms of the loss of taste, we can see the time to sustained alleviation for both 100 and 200 milligram groups, they have a statistical difference. And for both of these groups compared to placebo, there is a statistical difference in time to sustained resolution of loss of taste. When we look at the significance of loss of smell and taste, clinically, we know that the SARS-CoV-2 can enter the central nervous system from the nerves of olfactory mucosa. So this has been proved according to past research. And in a lot of subsequent research, we know that the loss of smell is correlated to cognitive and emotional impairment in subjects with COVID-19. And in these long-term loss of smell from COVID-19, actually, basically, which means patients with long-term COVID-19 symptoms, according to research, we found that they resulted in organic change in the brains. So we can see that the significant reduction in the duration of smell and taste loss suggests that SNS812 via aerosol in combination with early administration may reduce the risk of the virus invading the central nervous system through the olfactory mucosa. And there are other important exploratory endpoints, including the safety and symptoms that we talked about. We should look at the virus negativization time. So when they have done PCR 2 times and they've continued to be negative, we call it virus negativization. So we can see the 200 milligram compared to the placebo group has a statistical difference in the p-value. So even though so far we don't have a clinical trial that's comparing directly SNS812 and Paxlovid, but if we look at the past clinical trial data, we can see that compared to the placebos, compared to the placebo groups, whether it's the continued alleviation of all target symptoms or sustained resolution, in terms of the reduction of time, we can see SNS812 is can more effectively and rapidly improving the symptoms and mitigating the symptoms. Now, a quick sum up. Among the 14 COVID-19 symptoms, the SNS812 was more effective than Paxlovid in achieving sustained alleviation for 12 symptoms and sustained resolution for 11 symptoms. If we compare to the control group, the SNS treatment group shortened the time to sustained alleviation and sustained resolution of all targeted symptoms by 45%, with statistical differences. In contrast, when we look at Paxlovid, it only shortened these times by 7.6% and 6.3%, with p-values not reaching statistical significance. So compared to the control group, SNS812 reduced the time to sustained alleviation for 11 symptoms and sustained resolution for 12 symptoms, while Paxlovid only reduced these times for 5 and 8 symptoms respectively. Additionally, Paxlovid significantly increased the duration of diarrhea and nausea by 20% to 50%. The side effects prevented the assessment of these effects on loss of smell, loss of taste, and fatigue. So we are unable to evaluate it clinically, but we can see it from our results. So compared to Paxlovid, we can actually achieve clinically proved improvements. And in terms of the main primary endpoint, SNS812 has shown no drug-related adverse effects. In terms of the secondary endpoints, which is the key efficacy endpoints, the time to resolution of all COVID-19 signs and symptoms, time to resolution of shortness of breath related to severe cases, time to resolution of neurological COVID-19 signs and symptoms, and time to COVID-19 virus negativization. So this is a result that we wish to see when we design this clinical trial. So it means that inhaled SNS812 provides the best treatment option in COVID-19. That's my presentation on the progress of our clinical study and the progress that we've seen and the results that we've seen. Next, we would like to invite Dr. Zhang Shuyuan of the Department of Clinical Laboratory Sciences and Medical Biotechnology at NTUCM on the analysis of virus strains in the subjects. Good afternoon, everyone. I am Dr. Zhang Shuyuan from National Taiwan University. I would like to talk to you about the analysis of virus strains in subjects in this phase 2 clinical study. According to the study that was shared, we've recruited 135 subjects. The viruses from these subjects were all successfully sequenced and divided into their genotypes. They were divided into 8 most prevalent genotypes, including close to half of them were JN.1, the most prevalent, and including the KP1 to 4, which began to be prevalent in Q2 this year, and even the newest and highly contagious strain LB.1, accounting for 17.1% of the findings. Overall, we can see from Q4 last year to Q2 this year, the enrollment period of our clinical study, the virus strains observed are consistent with the variants that were prevalent during this specified period. We must know that the continued variations and mutations of the viruses, it started from Omicron to BA2, and next strain analyzed these virus strains and looked at how high their immune escape are. So we can see the red are higher in immune escape of the virus strain. So we can see the SNS812 enrolled subjects covered over 90% of the high immune escaped strains. Why are we talking about these high immune escaped strains? Because when we first started being vaccinated, those were wild types. But in 2021, we found Delta strains. We realized 2 vaccines were not enough. We needed 3 to 4 booster, a third or fourth vaccines as boosters. And then there were Omicron, and we see how, so we have the double potency vaccines, which is the wild type plus Omicron BA1 or BA2 vaccine strains. But despite this, we could not prevent the mutations of these viruses. So last year, there was the XBB 1.5 booster. And early this year, we saw JN1 strain. So when we look at from Omicron to JN1, we can see the receptors increased from 32 to 58 mutations. And in this graph, we can see that the mutations on JN1 not only increased its contagiousness, but also its ability for immune escape. According to the latest study, we can see that they analyzed patients that were infected with XBB 1.5. We can see that even though these patients were infected with XBB 1.5, there was 5 to 6 fold decrease in neutralization against JN.1. So we can see it's mutating very fast. And early this year, the FDA suggested that they will target JN1 for vaccines for this fall, but in a few days, the FDA then updated the substrain as KP2 for COVID vaccine. So we can see the virus mutation is happening very fast. So with these quickly mutating variations, we can see that the SNS812 are targeting a group of genomes or the sequencing that has remained consistent throughout all the variations, which means this drug will remain effective despite the variations of the virus strains. So in summary, the SNS812, because it specifically targets the enzyme location of the genome, so it's a very specific drug. It can safely and effectively inhibit broad spectrum variants. The target genome sequence has remained unchanged since the emergence of SARS in 2003 until JN1, which has the strongest immune evasion discovered so far. So it's broad spectrum, and because of this mechanism, we can see that it led to the effective efficacy as shown in phase 2, which means SNS812 is a safer and more precise treatment. Thank you very much, Professor Zhang Shuyuan, for your presentation. Next, we would like to invite President of Oneness Biotech to share with us SNS812 outlook and future prospects. Thank you.
对我们
Thank you for Dr. Chen and Professor Zhang to explain and report our phase 2 results and introduce us some COVID-19 knowledge. We hope in the future I want to share with you the SNS812 outlook and future prospects. To sum up, I believe you all know a lot of influenza, so I want to make a comparison between COVID-19 and influenza. COVID-19 is actually have a higher infectivity than influenza, and the estimated infected population each year is expected to be more than 110 million. The mortality rate is also higher than influenza. It's about 4 times higher than influenza. And about the estimation of medical markets for influenza, it is estimated to earn about 6.4 billion to 2030. But for COVID-19, the profit is estimated to be 16.2 billion to 2031, which is much higher than influenza. And about the sales figures, the Paxlovid accounting for 70% of the market share. In the first half of 2024, the sales revenue is about 2.3 billion. And in terms of influenza vaccine, it's between 40 to 4 to 9 billion, depending on the influenza pandemic. That's our market status quo. We can see the market prospect for the SNS812 because COVID-19 is not seasonal and its cycle is much shorter than influenza. As mentioned before, we have like 3 waves of pandemic each year, whereas the influenza only has 1 wave of pandemic. And the COVID-19 positive rate at 12 rate is 3 to 4%, which is also much higher than influenza, not to mention its much higher mortality rate. So the base number of COVID-19 will be definitely higher than influenza. That's why we have a higher outlook for COVID-19 market than influenza. So the Oneness Biotech and Microbiome Shanghai will cooperate under this excellent foundation. We will continue our work to negotiate with many governments and international pharma companies to initiate phase 3 studies. And we will also discuss with other regulatory authorities for EUA application. We will try to make our drug as an emergency usage medication for other countries. Thank you.
Thank you very much to our 3 experts and 2 presidents for your presentations and your QA. We would also like to thank all of the friends of the media and investors in person and online. In the interest of time, we are unable to answer every single one of your questions, but we will answer the questions in written form on our company website after the press conference. We now invite the press for a joint interview. This is the end of our press conference. Thank you so much for your participation.
谢谢大家。